Hello, everyone, and welcome to today's live broadcast, Scaling Cell Therapy Manufacturing, presented by Jim Beltzer, PhD. We are excited to bring you this educational web seminar presented by LabRoots and sponsored by Terumo BCT. Terumo BCT is a global leader in blood component, therapeutic apheresis, and cellular technologies. It is the only company with a unique combination of apheresis collections, manual and automated whole blood processing, and pathogen reduction technologies. Terumo BCT believes in the potential of blood to do even more for patients than it does today. This belief inspires the company's innovation and strengthens its collaboration with customers. To learn more about Terumo BCT, please visit www.terumobct.com. I am Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. This webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window. Type your question into the box that appears on the screen, and we'll answer as many questions as we have time for at the end of the presentation. Also, please notice that you'll be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located on the lower right. And finally, if you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or use the Q&A button to let us know you're having a problem. Thank you. Now I'd like to introduce Dr. Beltzer. Prior to joining the cell processing team at Terumo BCT, Dr. Beltzer served as lead scientist for the R&D of Corning Synthemax, a synthetic growth surface for the culture of human embryonic stem cells at Corning Life Sciences. He was a co-founder of RET Corporation and was a key player in the industrialization of several technology platforms, including phage display and yeast-2 hybrid. He has more than 20 years of industrial research and development experience in a broad range of disciplines. Please join me in welcoming Dr. Beltzer. I will now turn the presentation over to him.
Today I'd like to present 3 case studies in cell therapy manufacturing. T-cell expansion case study, viral vector production case study, and finally producing a 10 quantum system lot. Before I get to that, I would like to introduce you to the quantum cell expansion system. The quantum is a novel hollow fiber bioreactor system that is closed, uses continuous perfusion, and is a single-use system. It's primarily designed for adherent cells, but we also have some results growing suspension cells in the quantum. We have automated all of the necessary steps for traditional cell culture, and we have extremely good control over the gas exchange and the temperature in the quantum. This is the single-use part. This is the quantum disposable, and you can see the bioreactor on the right, and then the tubing organizer is how the plumbing interacts with the pumps and valves on the quantum system. And then to the left of the screen, you can see a couple of the bags that are attached to the harvest bag and the waste bag. So here we have the quantum system with that disposable mounted. So you can see the hollow fiber bioreactor in the middle of your screen. You can see the 4 pumps that control the fluid input to the system and the circulation of the fluid in the system. Also on the left, we have the gas inlet and the gas transfer module, which is responsible for all of the gas exchange that takes place in the quantum system. So where the rubber hits the road is the hollow fiber bioreactor. It's composed of about 11,500 semi-permeable hollow fibers, and you can think of those as very small straws. The equivalent surface area is about 120 T175 flasks. And the cells grow on the inside of those hollow fibers, as shown in the micrograph that's on the top right of the screen. So there are basically 2 different fluid paths within the hollow fiber bioreactor. The IC loop, which runs down the center of those straws of the hollow fiber and basically passes straight from left to right through the center of the bioreactor. And then there's the extra capillary loop, and you can see the inlet and outlet of that sticking up on the top of the bioreactor. And that's basically the bathwater that fills the space that surrounds these hollow fibers. And so there we can achieve perfusion feeding of the cells. We can use both of these loops to our advantage. So I show here a hydraulic layout system for the quantum with the IC loop and the EC loop shown in red and blue, and the hollow fiber bioreactor shown in the middle. So these controls of inlet and circulation pumps will become very important. So with the door closed, the bioreactor is now an incubator. In fact, we call it a clean room in a box. All of the controls are accomplished through the automated touch screen on the front of the machine. You can see running across the top, the inlets and outlets are described, and that's one thing. The quantum's not smart enough to know which bag you've attached to which inlet line, so you have to call that out. You also have control of the various inlet pumps on the yellow or for the IC side, and in the blue or for the EC side, and you can see the bioreactor in the middle of your screen. Now, our engineers have gone to a lot of trouble to describe a number of predefined tasks and multi-step tasks, so they're either single-step or multi-step. These describe, to the best of our ability, all the necessary tasks that you need to perform to culture cells in the bioreactor. But the most important part of this entire slide is the purple button that sits above each task description, and that's the modify button. So all of these tasks are modifiable to meet your cell culture needs. In fact, you can create custom tasks and link them together to describe even as much as your entire run in a single custom step. We track the growth of our cells by sampling media, most commonly from the EC port. There's a Luer lock port that's shown on the left. But if you need to get a sample of the contents of the IC coil, right, so that's the IC loop that the cells are growing on in that loop, you can weld off a portion of the sample coil as shown on the right. Before I go into the case studies, I'd like to introduce a CAR T-cell therapy manufacturing that's in development at Terumo. So the basic process is an apheresis product can be purified into lymphocytes, and then those lymphocytes are either gene modified and then activated and expanded. Finally, the expanded cells are concentrated and washed and eventually infused into the patient. This can be accomplished using Terumo devices, so the Spectra Optia can give you apheresis product, and lymphocytes can be purified using the Elutra cell separation system. So I'd like to concentrate on two aspects of this process that utilize the quantum system. The first is activation and expansion of T cells in the quantum system. And the second is the production of viral vectors using the quantum system as well. So let's start with T-cell expansion in the quantum system. So this is our basic cell expansion of T-cells in the quantum system, and we typically start out with 2.5 mils of a leukapheresis product or 25 mils of an elutriated product, and you can get those either from the apheresis or from the Elutra machine. We loaded 100 million lymphocytes into the machine, approximately 228 million, of those cells were CD3 plus T cells. Those cells were activated with CD3, CD28 Dynabeads in a 2-to-1 ratio of cells to beads, and then fed by continuous perfusion. So that's on day 0. On day 3, 6, and 9, we introduce a bolus of IL-2 and IL-7, as you can see on the timeline. And there's a minimal amount of circulation in the IC loop where the cells are of 1 ml per minute, so that's really quite slow. And our gas exchange is taken care of by circulating the EC loop at 100 ml per minute. That's also clearing out any waste products that may have been produced by the expansion of the cells. After day 6, we do flip the bioreactor once a day just to break apart the clumps. Finally, on day 12, we harvest the cells, and we were able to harvest, after a 12-day expansion, about 15 billion T cells, greater than 500-fold expansion in 12 days, and in a low harvest volume of 0.5 to 1 liter. Now, I've shown this same data in a different format here. So you can see on day 0, we started out with 28 million CD3 plus T cells, and then in the blue bar in the last column, we end up with 15 billion CD3 plus T cells. Also of interest is that we get a selective enrichment of the CD8 cells that shows up quite nicely in day 9 and day 12. If we look at the flow results, you can see that we end up with a 99% pure T cell population, and that's also a preferential expansion of our CD8 cells, as I mentioned before, coming up to about 69% in the final product. There's a significant drop in the PD-1 expression, and our T cells are expressing IL-2 and interferon gamma, the activation markers. So just in summary then, we had over a 500-fold expansion of CD3 plus T cells over 12 days. No pre-processing, a closed system, automated, reduces labor, it can be used in a controlled space, perfusion fed, and we use a low amount of media in the process, and we have a low volume for our harvest. Moving on to viral vector production in the quantum system. So many of you are familiar with growing incubators full of flasks or cell stacks or roller bottles in order to collect your viral vector supernatant. And I've heard stories of up to 16 10 stacks going into a single prep. So this is a labor-intensive process. Many open steps. There's a clean room is required because you've got high concentration of virus, large volumes of vector, and it's very contamination prone. There are two types of virus expression. There's stable and transient expression of viral vectors. Stable transfection of retroviral vectors is usually done using a packaging cell line, and your gene of interest is introduced into a packaging cell for those purposes. Transient expression, on the other hand, involves introducing all of the genes required to produce the virus, including the envelope, the GAG, and the Pol genes. So you would introduce a mixture of 4 plasmids into the transient transfection mix. Both the packaging cell line and the transient expression systems have their pros and cons. So the packaging cell line is obviously very convenient and allows for stable expression for weeks, but that initial construction of the cell line can be difficult. Transient expression, on the other hand, is stable for 40 to 72 hours, and it is fast and achieves comparable titers. The cons, it's batch-to-batch variability and difficult to scale. Both of these protocols have been used on the quantum system. Here's a cartoon of our transient expression experiments to produce lentiviral vectors in the quantum, and this was done in collaboration with Gerhard Bauer's lab at UC Davis Regen Medicine Center. So the first is to load your HEK293 T cells. If you're in a hurry and you want to start the next day, then you would load at least 1 billion cells into the bioreactor. If you wish to let the cells expand first in the bioreactor, you can load fewer cells and allow them to expand. So cells are loaded and allowed to attach to the bioreactor for 15 hours or overnight. And then when the population of cells has filled the bioreactor to approximately 1 to 1.5 billion cells, then you can begin your transfection. And so that's day one of the process. So the transfection, you add the 4 plasmids, as I just spoke about, for the transient expression of lentivirus in calcium phosphate, and you allow the transfection to proceed for 15 hours in the quantum. And then at the end of that 15 hours, you wash out and do an induction for another 9 hours. So at the end of that time, we want to collect the virus. This is on day 4, and we wanted to be able to concentrate the virus afterwards, so there was a switch to serum-free media, and we then proceeded to feed the cells for 40 hours. During that time, we continuously collected the viral vector in the supernatant, or the conditioned media that passed over the cells. On day 5, we harvested the cells. We got back our 1 to 2.5 billion 293 T cells. We were able to titer and concentrate the virus particles and determine the MOI and the TUs per mil. So we've shown that the quantum system can be used to produce retroviral and lentiviral vectors. It's automated. It's capable of achieving both stable and transient expression systems. It's a closed system. The vector concentration is equal to what we see in flask-based systems. And using engineering controls, we can control the volume of the vector containing supernatant. And in the next slide, I show the hydraulic system layout again to show you how important that is in concentrating and decreasing the volume of our virus containing supernatant. So basically that's achieved by harvesting the IC loop. So you can see in the diagram that we harvest the IC loop into a separate bag from the EC loop where we accomplish the feeding of the cells. And so in summary, the results of our viral vector production in the quantum, for the lentivirus, we got about 2 times 10 to the 8th viral particles per mil, which was a similar yield to what's normally obtained in the cell stack. Likewise, gamma retrovirus gave similar results at UFETs to what they'd been getting in their plastic-based system. And there will be references for both of these results at the end of the talk. And moving on to the allogeneic model, which is a case study in scaling out the quantum system from 1 machine to 10 machines. So this case study was a collaboration between Athersys and its European subsidiary Regenesis and the contract manufacturing organization of MasterCell. So the object was to transfer the process, so growing MAPCs in the quantum, to scale that process out from 1 machine to 10 machines to create a larger lot size, and then finally to see if there were any efficiencies that could be gained. And cutting straight to the chase, we found that there was a 40% cost, or 40% reduction in the cost of goods producing cells in this way. So in the first slide, we show the technology transfer timeline through to scale out. So the first few weeks were spent simply learning how to run the quantum. And then Another 6 weeks were used for process optimization. Most of this work was on downstream processing of the cell product. And finally, the 10 quantum runs were done in the scale-out. So this is a comparison of running a single quantum system here in the box on the bottom. It tells you how many people are required. So you can see on day 1, we take 2 operators to run the quantum system. That's mostly to prepare the cells and the quantum for loading. With a 10 quantum system, you see at the box at the top that 2 operators are also required in day 0 and day 1, or day -1 and day 0. But basically, the intermediate steps can be handled by a single operator. And as I said before, the harvest day is the most difficult, and here we needed more help. So finally, if you look at the product that we got, you can see that the flow cytometry profiles from comparing the quantum-produced cells to the cell stack-produced cells are equal, and the same is true for the protein secretion. So you can see that we were able to develop a process that can yield consistent cell quality and yield, and can simplify the progression from learning to production. And importantly, there's a 40% cost reduction in cost of goods. And we were able to produce large lots of cells. So here are the references for the lentiviral, the retroviral, and finally the scale out of the quantum cell expansion system. Thank you very much.
Thank you, Dr. Beltzer, for that informative presentation. It is time for Q&A. If you have a question you'd like to ask Dr. Beltzer, please do so now. Just click on the green Q&A button at the lower left of the presentation window, type your question into the box that appears on your screen, and click on the send button. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, can you control the shear stress in the quantum for the growth of endothelial cells?
Thanks for that question. First of all, there's very little shear stress in the quantum system. That's pure physics. If you've got a 1/4-inch inlet line going into the IC loop and a 2-inch in diameter bioreactor, there's very little shear that happens. Having said that, though, we can control the flow rate of the media through the IC loop with our pumps, and if needed, we can even feed across the membrane without creating shear stress.
How can I monitor the growth of the cells in the quantum?
So you can monitor the growth in the quantum by taking a sample from the EC sample port. So that's a Luer lock port that is sterile, so there's a 0.2 micron filter behind it. We recommend that you monitor the lactate production and glucose consumption, and typically those measurements are done once every 24 hours.
Has the quantum been used to culture iPS cells?
Yes, the quantum has been used to successfully expand iPS cells. We haven't done differentiation of iPS cells yet in the quantum, but that's ongoing. In fact, recent experiments have shown that the quantum can significantly reduce the media required to expand iPS cells.
What is the media perfusion rate in the quantum?
Well, the media perfusion rate typically starts out at 0 or very low, so 0.1 mil per minute in the beginning stages of a typical expansion, and it may go as high as 1.6 mils per minute at the end of a typical MSC expansion. For viral vectors and other applications, we may have to go higher as the cell densities increase within the quantum. So for the viral vector case, we were running up around 2.5 mils per minute.
Is there a reason for switching to serum-free media for viral vector production?
Yes, so that's a good question. If you produce your viral vectors in serum-containing media, it complicates the concentration of that virus product after you have finished your run. So the serum proteins would quickly clog the membrane concentrators that are typically used to concentrate viral vectors.
How are the Dynabeads loaded into the system for T-cell expansions?
Very simple. We basically mix the Dynabeads with the cells in a cell inlet bag prior to loading the cells into the bioreactor. And as I mentioned, the ratio is 2 to 1.
What kind of media is used for T-cell expansions?
To date, we've tried both Tex-Max and RPMI plus 2% human AB serum. And right now we get the best results with RPMI, but we plan to test more media in the future, so stay tuned.
We're almost out of time, so this will have to be our last question. How are suspension cells kept in the hollow fiber bioreactor?
So our pumps, the circulation pumps in the quantum cell expansion system can be reversed. So we can run those pumps in such a fashion as to push any cells that may have gotten out into the rest of the IC loop back into the bioreactor. So again, we just use our circulation pumps.
I would like to once again thank Dr. Beltzer for his presentation. Do you have any final comments?
I just want to thank everybody, and if you have any questions further to this, please contact me at my email that should be on the last slide of the presentation.
Well, thank you once again. I would also like to thank our sponsor, Terumo BCT, for making today's educational webcast possible. This webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. And before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through November 31st, 2017. You'll receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us, and we hope to see you again soon. Goodbye.